Hey, hey, hey! This PowerPoint is designed to help you with your second exam for microbiology at Gaston State Community College. Let's go ahead and jump in. Okay, the idea is how do bacteria, very similar process at the cellular level to what we do, but how do bacteria break down food and then build what they need? And so bacteria, just like humans, have to have a food source. So without food, they're not going to survive. And so that is a little bit encouraging if you think about infection. So how long will the organism survive on a countertop? And different organisms survive different amounts of time, but they're not going to survive long unless there's food and water. You know, just like a human, they're not going to be able to survive for an extended amount of time on a vending machine button. Things like that. They're not going to be able to survive without a source of food. So think about where bacteria most frequently grow. It's places with nutrients and water. And so anyway, the word metabolism is really two different things. Catabolism is the ability to break down, while anabolism is the ability to build up. So, you know, whatever you ate for breakfast, you're breaking that biscuit down. That's catabolism. And then in our bodies, we've got to build DNA, so do bacteria, and then we've got to build the proteins that we need, you know, hormones and things like that we need to build, that we have to survive. And so bacteria are similar, much more simplistic, but similar to that. They have to have a food source to break down catabolism, and then the picture shows putting it back together in maybe in a different order, proteins and things like that, for a food source. So one thing I want you to notice right off the bat from this image, not really notice it from the image, but just make a note of it. Right here, this bond is broken. So imagine that these are like carbons and hydrogens, whatever. Right? If you've taken some organic chemistry in your past, when this bond right here is broken, and it looks like they're all broken actually. So they're broken into those little components, those small pieces. That's catabolism, breaking large molecules into smaller ones. When the bonds are broken, energy is released. So that's where we get our energy from. You know, be it chocolate or caffeine or carbs or fat or whatever, the energy is released when the bonds are broken. And so different bonds release different amounts of energy. And then anabolism is where we're going to take all these little pieces. We're going to take all these little pieces and put them back together. That's the buildup. Taking small particles and putting them, building larger particles, polymers. And that requires energy. So to put things together, like to build a segment of DNA, requires energy. It's actually very energy consuming. Okay, so that's what this slide is going to tell you. Metabolism is the sum of the chemical reactions. There's anabolism, simple substances combine to form complex substances. This requires energy. And then catabolism is where it's broken down. Energy is required. Okay, most bacteria, all protozoan and all fungi are what we call but I didn't do very good on that, did I? Chemoheterotrophs. That means we consume some form of chemical, like a sugar, some chemical, mainly sugar. And then heterotroph, that word, you may know that word from a previous lecture or science course. The word heterotroph means that we consume food instead of making it ourselves. The opposite of heterotroph is autotroph, and that's what plants are. Plants don't have to consume food. They can make their own food, and then they break it down. So a chemoheterotroph, you want to know that. Most bacteria are chemoheterotrophs, which means they use a chemical such as glucose for energy. Okay, this picture, real simple, just showing you ATP is energy. So from this point forward in your life, know when you see the letters ATP, that's energy. It just shows simple molecules are anabolized and then catabolized. Break down, put together. Break down, put together. Okay, so we're going to talk today about three different ways to break down a sugar molecule. We're going to talk about sugar, a sugar molecule to get energy. So three different pathways. And bacteria, like animals, do one or the other. And that, we have muscle cells that can do two different ones, but for the most part, we do one or you just choose one, and that organism does one. So this is three different ways that we can catabolize, break down a molecule of sugar to get energy. You got to have energy. Bacteria have to have food and water so that they can have energy. And without energy, they're dead. Same with us. Okay. Easy breezy. All three of these, we're going to talk about the steps of them. All three of these, 1, 2, 3, start out with the first step called glycolysis. And the word glycolysis, break sugar. That's what the word means. And so this is the formula for sugar. And you're going to break it apart. And when we do, you get 2 pyruvic acids, which are basically, and it's really a long process, but all you really want to know for this class is that, and really for all classes, I think, is that when you take that glucose, it's got 6 carbons, 1, 2, 3, 4, 5, 6, and they're bonded together. When you take and you break that apart, right there, you get 2 pyruvic acids. And so that's what happens in glycolysis. You get 2 of those pyruvic acids. And I don't know if you can see that good because the water thing on the bottom. Anyway, 2 pyruvic acids are made from 1 molecule of glucose. And we call that step glycolysis. Okay, so aerobic respiration, anaerobic respiration, and fermentation. Those are our 3 things we're talking about. So the first one, look at the steps. Step 1, glycolysis. Step 2, the Krebs cycle. Step 3, the electron transport chain. So that's just the name of the 3 steps in aerobic respiration. Okay, you really want to know the details after that. At the conclusion of aerobic respiration, from 1 molecule of glucose, right? We did glycolysis, Krebs cycle, electron transport chain. You get 36 or 38 ATP, right? So that's key. Somewhere between 36, that's for eukaryotes, that's cells with a nucleus, everything but bacteria, that's us. Bacteria get 38 molecules of energy. Okay, and you have to have oxygen. Look at the word, aerobic respiration, oxygen. That's key. That's why I underlined it so many times. Oxygen is required. That's how you and I get energy. We eat a biscuit from Chick-fil-A, chicken biscuit from Chick-fil-A. You eat that biscuit and break down that biscuit using oxygen. You got to have oxygen. You got to have food. And that's how you get energy. And you have to have energy for your heart to pump, your lungs to expand, your muscles to contract, your brain to function, all of that. So this is the most efficient way. These are listed, all 3 of these are listed in efficiency. So aerobic respiration gives you, maybe write it in somewhere, the most energy. Right? That's not very good. I'm not doing very good. There you go. The most amount of energy is from aerobic respiration. Okay? Moderate amounts of energy is anaerobic respiration. Same 3 steps. Easy breezy. Same 3 steps. Glycolysis, Krebs cycle, electron transport chain. The major difference is no oxygen and moderate energy. Somewhere higher than 2, which is what fermentation is going to give you, but less than aerobic respiration. So this is medium. This is your moderate. Okay? No oxygen. There is an electron acceptor. That means it's going to catch that final electron. The bonds, when they're broken, they have electrons in them. So when that final bond is broken and the electron is released, it's not oxygen. It's something inorganic. That's key too. This oxygen is also inorganic. Does it say that? Write that in. Oxygen is inorganic. Okay. something like a sulfate or phosphate, something in the soil that they can use other than oxygen, because this is without oxygen. And then perhaps the most interesting, gives you the least amount of energy. So this is the least efficient. It does glycolysis. Remember, when you break apart the glucose, you get 2 molecules of pyruvic acid or pyruvate. In order for fermentation to start again, they just have to take that pyruvate and change it into something else to start the process again. And so they convert the pyruvic acid into lactic acid fermentation, right here, or alcohol fermentation. So the only energy you get is from the 2 ATP. So 100% of energy is from glycolysis. Glycolysis. Sorry. Okay. All of it is from glycolysis. That's it. Alcohol fermentation is just the other option. It can convert that pyruvic acid into lactic acid, or it can do ethyl alcohol. Okay, so that's the final electron acceptor. Both of these are organic. Okay, aerobic and anaerobic both had inorganic. Key, know that. Fermentation has an organic electron acceptor. So when the final bond is broken, the electron is released. What catches that final electron is going to be organic, lactic acid or alcohol. This is the beer and wine and bread industry. Alcohol also releases the CO2. That's what makes the holes in bread. That's what makes bread rise. Lactic acid, that's how we work out. If your muscle cells hold a certain amount of oxygen, and when you have used up that oxygen, let's say you're working out a little bit harder than you normally work out, or jogging a little bit further or faster, you've used up your oxygen, then muscle cells, for a small amount of time, not forever, can do lactic acid fermentation. Small amount of time. They can't do this forever, but they can do that, and that's what makes you sore after working out. So those two are, it's lactic acid fermentation or ethyl alcohol fermentation. Okay, so just a recap. 3 different ways to catabolize, or to metabolize, break down and get energy, was aerobic. Oxygen is required. You get anywhere from 36 to 38 ATP. The oxygen is inorganic. Steps: glycolysis, Krebs cycle, electron transport chain. With anaerobic respiration, same 3 steps. Oh, it's going to make me go all the way back. Same 3 steps. Final electron acceptor is inorganic, and it makes a moderate amount of energy, somewhere higher than fermentation 2, but less than aerobic respiration at 36 or 38. So less than aerobic respiration. Okay, and then we talked about fermentation makes only 2. All of the energy produced comes from glycolysis. Okay, next topic is really talking about, kind of not really the next, it just kind of moves into, with fermentation, we talked about a little bit about beer and wine. And so what determines your outcome? What determines whether you get vodka with potatoes or whether you get bread with yeast and those ingredients? Okay, so the end product of fermentation depends on what organism did you use. That's right here. What organism did you use? Did you use, the most common is Saccharomyces. That's used for alcohol and bread. You can buy it in the grocery store in the little packets. What organism did you use? What did you feed it? Did you start with flour and sugar? Did you start with potatoes? Did you start with wheat or strawberries? What is the substrate that's used? And then the enzymes that are present in that organism. So you want to know those 3 things. The organism, the substrate, and the enzyme. Saccharomyces is the most common. So this is a table out of your book. You do not have to memorize this. This is not on the test. It's just for an example. So if you start out, this is just example purposes. If you start out with Saccharomyces, that's a yeast, most common is Saccharomyces, and you feed it, that's your substrate, malt extract. With the enzymes that are present, you can make homemade beer. This table is backwards. I think it should read the other way. Okay, if you start with just some interesting ones, Lactobacillus is a bacteria, and you feed it cabbage with the enzymes present in the Lactobacillus, you're going to get sauerkraut. You can see Propionibacterium bacterium, lactic acid gives you Swiss cheese. So this was just for example purposes and interesting. You can give, some sort of fruit with a certain organism and make different types of wine. So anyway, this was just interesting. Okay, so 2 reasons we really care about enzymes. Now in class we're going to do some cool, short, interesting enzyme tests. And that's really why we care about them for the health side, for the medical side. You can take and test, is your infection strep or staph? Take and see, is it E. coli, things like that. So we're going to do some simple enzyme tests in class to go along with this lecture. But we care about them in this class for growing bacteria. So to grow bacteria for study and to help us identify and distinguish, is it strep or staph? Is it E. coli or Propionibacterium or whatever? Okay, when we talk about enzymes are proteins. They're made up of amino acids just like proteins are, but enzymes have a specific job that they do. Okay, so, for example, there's an enzyme in your saliva that helps break down carbohydrates, and that enzyme is called amylase, and that's all it does. It's called salivary amylase, and it breaks down carbs. If you eat carbs, it starts the breakdown process. You have another one, gastric amylase, in your stomach. If you've taken 202, you remember a lot of those digestive enzymes. They have a job, right? Lipase, most enzymes, not all, but end in the letters ASE. Okay. So enzymes, they eat a certain substrate. So enzymes act on substrates, okay? And that's really all I'm going to mention about this picture until a little bit later. This picture should be later, actually. Okay, so this is just some facts about enzymes. All organisms, including bacteria and us, have enzymes to lower the amount of energy, to lower the amount of energy required. Okay, so digestion is easier, requires less energy because of enzymes. So without the enzymes, it would be a more consuming process. Enzymes are specific. They act on their substrate. The textbook used to say that it fits together like a lock and key, but now the analogy is more like a glove and hand. They fit together and mold together like that last picture showed us. The substrate fits the enzyme. The enzyme and substrate join together, look right here, at a site called the active site. So this is the active site. Can you see that? This is the active site. So the substrate and enzyme join together at an active site. And then once they join together and the enzyme may break or build the substrate, the enzyme is reusable. The substrate is forever changed. So after the enzyme helps bring about the reaction, the substrate is changed, but the enzyme, so insulin can work over and over and over. It can, or amylase in your mouth can break down carbs, break down carbs. The same enzyme can work on more than one carb. Okay, so here are things that would affect. So let's say that, I'm just making this up, I don't know the truth. Let's say that amylase can break down 10 carbohydrate molecules in a minute. I really have no idea, so I don't know if that's true or not. It might be 100, it might be 1. So, and we're just for example purposes. Amylase can break down a carb. We said 10 carbs in 1 minute. Okay, so these are 4 factors that would affect how well amylase can do that. So if the temperature isn't just right, it's probably going to do less. It's not, if it's above, if you have a high temperature, amylase is only going to work, and it can only do 5 in a minute, right? So increases in temperature that are too high look at this picture, will denature, break apart. The enzyme has to be folded just right to function. And so if the temperature is too high, next thing on that same slide, if the pH is too high or too low, then the enzyme will denature or unravel. Okay, so it said, so increases in temperature. You think, oh, if I wash my dishes on hot or I wash my laundry on hot, it'll work better. Not necessarily. Increases in temperature that are too high will denature the enzyme and prevent the reaction. So temperature does not always increase the reaction. pH, same thing. Most work best at a neutral pH between 6 and 8. Changes outside of that will denature. So not all, but some work best, let's say at 7. The pH of human blood is 7, right around 7, 7.2, 7.4. So the enzymes in human blood work best at that pH. The ones in the stomach work best at 2. So the optimum pH may change, but outside of optimum for that specific enzyme, the enzyme will denature. Okay, the amount of substrate. Amylase, we said, for example, amylase breaks down 10 carbs in a minute. Well, not if you're not eating carbs. Not if you only ate celery. Celery does have some carbs. But anyway, you get the point. If you only drink water for that hour, then it's not going to break down 10 every minute because it can't. And then 2 types of inhibitors. Let me go back to that initial picture. You keep looking at the words. Okay. There's 2 types of inhibitors. There's a competitive inhibitor and there's a non-competitive inhibitor. So looking at the picture, the blue one is a competitive inhibitor. It bound to the active site. Remember, that's the active site. Right? Remember, this is the active site. So that blue inhibitor bound to the active site, blocking. So if the inhibitor is there, then the substrate can't bind, can't make 10 in a minute. Okay. A non-competitive inhibitor is this yellow one. It binds to a site other than the active site. It's called an allosteric site, but you don't need to know that for your test. It binds to a site other than the active site, changing, look how this has changed shapes, if it's supposed to be like that. Changing the shape of the active site. So both of these are going to slow down the rate of the reaction. So you used to do 10 a minute, now you can only do 3 or 4 a minute because these inhibitors are in the way. Okay, the one you want to know for sure is a competitive inhibitor. The example, remember what I told you, you always want to know the examples. That's what they're going to be on the test. An example of a competitive inhibitor is the sulfa drugs. Sulfonamide drugs is a type of antibiotic that works because it's a competitive inhibitor. So the enzyme inside the bacteria required to make folic acid, bacteria have to have folic acid. So the enzyme that produces folic acid, it breaks down a substrate called PABA. Sulfonamide is a competitive inhibitor. Eventually the bacteria can't make folic acid, which it has to have, so it dies. So let's look back at the picture real quick. Okay, so PABA is right here. Oh, no, I messed up. Sorry. It's this right here. PABA. Okay. PABA is the substrate. Okay. The sulfonamide drug is the competitive inhibitor. Okay. So when the PABA binds to the enzyme, it produces folic acid. Okay, so if you're taking oh man, I messed up. There we go. You know that says acid, right? Okay, if you're taking this drug, which is a competitive inhibitor, then PABA can't bind, so the bacteria can't make folic acid, so the bacteria dies. So sulfonamide drug is an example of a competitive inhibitor. Okay, that's where we were. Non-competitive inhibitors bind to the enzyme. So competitive bind to the active site, non-competitive bind somewhere else. This is showing you another picture. This, on the right-hand side, you can see a competitive inhibitor. This is the competitive inhibitor and the competitive inhibitor is sulfonamide. PABA is the substrate. They're generally shaped alike. So this just, you don't have to know this diagram or anything like that. Wouldn't ask that on the test. But I'm just showing you that they are similar. Fit together like a hand and a glove, right? This shows you non-competitive inhibitor. This just shows you denaturing. We mentioned that. Temperature too high or too low may denature. pH too high or too low may denature the enzyme. And once it's unraveled, it won't work. This is another example of just showing you the substrate and enzyme fit together, and then you make a product. Okay, and then this, if you were in class, I would stop right here, and we would do this lab, and then I'd go over the slide. But hopefully you've done it now. Okay, so you're going to take a microscope slide and put a little sample of strep bacteria and a little sample of staph bacteria. So strep and staph, you want to know this. Strep and staph, if you do a gram stain, they look the exact same. Coccus means they're round. Gram-positive. Strep and staph are always gram-positive. Always, always, always gram-positive. So they're going to be purple and round under a microscope. And sure, you're thinking, oh, but it's clusters if it's staph and chains if it's strep. True, in their natural environment. Well, honey, you took a scoop of it and smeared it around on that slide. So that's not their natural environment. You smeared them around. So you can't really tell if it was strep or staph. And chances are, most of your gram stains were like an enormous amount, just covered in purple circles. So you couldn't tell if it was clusters or chains. Okay, so you can't really tell the difference you cannot tell the difference in strep and staph with a gram stain. So this is an easy test. You take and put strep and staph on a microscope slide, put a dab of hydrogen peroxide, which would be the substrate in this story. If it's staph, it's going to bubble. If it's strep, it's not going to bubble because staph has an enzyme. We're going to test for certain enzymes. Has an enzyme called catalase, and catalase is an enzyme that acts on the substrate hydrogen peroxide, and it produces oxygen bubbles. Okay, so catalase bubbles in the presence of H2O2. The O2 is released. So you want to know this. Staph is catalase positive. Strep is catalase negative. So you're going to do several enzyme tests in class, but this is the only one that's actually on the test. It's the only one in the notes too. So by identifying if the enzyme catalase is present, we can tell if the infection is staph or strep. So a really easy way to tell. I hope this was helpful. Oh man, there's one more slide and I didn't know it. Sorry. I do still hope this was helpful. But anyway, so we already said that most bacteria are chemoheterotrophs. That's how I started right off the bat. Most bacteria are chemoheterotrophs. These are the other options. Okay, chemoheterotroph, we said, uses a chemical to produce, uses a chemical for an energy source. Heterotroph means that we have to consume something else. We do not make our own food. An autotroph means that they do make their own food. So plants and algae are photoautotrophs. They use light to make their own food. Chemoautotrophs use a chemical to make their own food. An inorganic compound. Photoheterotrophs, photo means light. Use light, but they still consume an organic compound. But they must have light. And then we already said most bacteria, that says M-O-S-T. Can you tell that? Because I have the free trial. That watermark is on the bottom, that Doceri desktop trial, but I'm sure you don't mind. Most bacteria. Okay, hope this was helpful.